Okay, we're going to get going in 1 minute. If you have the opportunity to do so and you have a mobile device that will allow you to take part in our surveys, we'll get going in about 30 seconds. So if you can get those out, that would be really useful. You're going to help write the final exam for me. So we'll do this together as a class. So I am going to talk to you today about a couple of different things. I'm going to go back and finish off the lecture that we had on memory. Memory itself, even though it's the most widely studied phenomenon in neuroscience, it's the best studied phenomenon in neuroscience, there are still a lot of questions that we have no answers to. And I'm going to take you through one of the current controversies that hasn't resolved itself. In fact, there was an argument at the last Society for Neuroscience, which just concluded last week, related to the same topic that I'm going to be presenting to you here today. And then I'm going to try to convince you at the end of today's lecture, and as we get into the next lecture, that in fact, some of the more higher order aspects of neuroscience are completely unknown to us. And as neurobiologists, we should be really interested in it. So before we get going, though, you're going to give me a hand with your final 2-hour exam. Non-cumulative, by the way, some of you have been asking. And if you can get out your phones or other mobile devices, and you're going to pick one of these folders for me. And on these folders, you have a choice that you are randomly going to pick related to the number of multiple choice test questions to begin with on your final exam. So choose wisely. Don't pick things right in the middle. So again, this is related to the number of multiple choice test questions that you'll have on your final exam. You are choosing the number of multiple choice test questions that I will put on the final exam for you. Okay, so it looks like it's a tie between, by the looks of it, between A and perhaps D. I think D is getting close, and maybe D as well. So shall we take a look at what you guys have chosen between? So this is the number of short answer questions on your final exam. If you have chosen A, you get 0 will choice all short answers. Not such a good choice. So if you had chosen B, which is not in the running right now, you would have had 15 multiple choice test questions. Okay, so I think the other possibility was C. And C, if you had chosen C, which looks like some people are now choosing, you would have had 20 multiple choice test questions. So I think we're going to stick with C. It seems to be the popular consensus of 20 multiple choice, which I will go through with you on the last in the last class, which will be a review. The other option was just 10. Okay, so we will go with, I think, 20 multiple choice test questions on your final exam. I will give you the precise breakdown of the number of questions that you will have per lecture on your final exam in the last class. All right? Hoping that's okay. You guys did that by committee. So now that you're familiar with this, we will go on to the next part, which is, again, of course, short answer questions. How many short answer questions do you actually want to write? There is no option of 0, by the way, on your Now you get to go. So you have a choice between A, B, or C. And it looks like the clear winner right now is D. So let's take a look at D first. And we're taking a look at the number of short answer questions that you're going to get on your final exam. It's not good news for your class, because it's 6. So you don't really want 6, right? 6 short answer questions, 1 page each. You really want 6? No. So how about we go back and rethink a little bit about what you want to do. So how about people who picked B? Split that between either A or C. And let's see if we get a clear winner here. And for time being, I think the winner is actually... Okay, are you a little bit nervous now? Because the numbers get bigger. Complete silence. Okay, so we'll go with A, and you will have four short answer questions. In fact, on the final exam, you will get a choice of 5 questions. You will choose 4 that you feel that you can answer well as short answer questions on your final. And then, of course, the last group. So now you get to decide how hard the final exam is going to be. So you get a choice of A, B, or C. It looks like it's pretty randomly split. That's a very wise choice, by the way. I don't know if you planned it that way, just to play it safe. You spread it out so that no one answer seems to be correct. So if you were choosing A, it would have been totally fair. So I think that was in the slight lead. Otherwise, your other choices would have been B, where it was going to be really hard. And do you think the last choice, what do you think? Really, really hard? No, I don't go with really, really hard. You either get fair, hard, or you get to... So we will go with a fair final exam with 20 multiple choice test questions. You will have about 10 one-line responses, and then you'll get a choice of 4 short answers where you will have 5 questions presented to you, but you will choose 4 out of the 5. You don't have to write all 5. It's 2 hours, and I'm hoping that that's going to give you enough time to be able to review your answers and everything else, okay? So thank you for taking part in that, and thank you for helping go through the final exam. I will post those details for everyone just to be really clear on that on your announcements page. And I will also, in your last lecture, where it will be an in-class review, also go through the exact breakdown of the number of questions and everything else on that review for you related to multiple choice, etc. So we were sort of left with this choice here in the last lecture, which related to looking at Todd Sachter and this individual here, Rick Hugener. These are actually 2 really big names in neuroscience, and they're embroiled right now in one of the biggest controversies in neuroscience related to learning and memory. And this is actually a really serious issue because the outcome of what happens to learning and memory, as determined by what's happening with these 2 individuals here, really determines what you learn and what we think learning and memory is all about. So I sort of went back and forth a little bit, and I didn't cover these slides, just to give you a little bit more context. In the last lecture, we learned about this phenomenon that is incredibly well studied, and we know that it's probably true with about 99% accuracy. We know that LTP, the long-term potentiation or the strengthening between synapses, is probably a phenomenon that takes place early after these 2 synapses communicate with each other, or neuron A communicates with neuron B. We think that that process of LTP strengthens the synapse, but that is not the end of how you remember something. That is not the storage of memory. And we know that this can't be the storage of memory, and the storage of memory involves a different mechanism that we're going to call consolidation and the formation of memory engrams for a variety of reasons. And in fact, we know that it has to go outside of the hippocampus for a variety of reasons that I'll go through in a few moments. So when we look at systems consolidation, where you take a memory that starts off in the hippocampus, remember sensory information is coming into the hippocampus, visual information, auditory information, you get the strengthening of synapses. You're saying here right now, some of this is being strengthened in your synapses right now at this instant. But you're not going to be able to form a memory that you're going to be able to draw on 1 week from now if it stays in the hippocampus and only in the hippocampus. And so we know that the synaptic strengthening has to be mirrored, and this memory trace, or this group of synapses, actually has to sit outside of the hippocampus and in other parts of the brain. And this, again, is very controversial in its own way. Many individuals here at U of T have contributed to our knowledge and our background knowledge of what consolidation is, moving short-term memory into a long-term storage outside of the hippocampus, which is what we're calling consolidation, actually has been pioneered by individuals like Dr. Morris Moscovich in psychology, as well as Dr. Paul Franklin, also in psychology and physiology. So we have to consider, how does this short-term memory process that starts in the hippocampus How do we actually get it into the cortex or into other areas of the brain? And this consolidation is actually something that we are going to be looking at today. We think that consolidation results in a very, very long-term form of memory. So something is brought into your hippocampus, sensory information is brought into your hippocampus. Those synapses that are taking part in this sensory information coming in are getting stronger. They're undergoing LTP. Those synapses will indeed become stronger. If you're learning what's up here on this slide right now, if you're thinking about what's up here on this slide, but that's not the end of it. At the end of it, parts of that information, if you're going to store this for longer periods of time, have to leave the hippocampus. It starts here, begins here, it continues to stay here, but it also has to start to leave. We know that there are pathways that leave the hippocampus. For example, we know that the fimbria fornix forms these axons, not the Schaffer collaterals, the CA1 neurons will then, their axons will form this fimbria fornix, which will then lead to other areas within the frontal cortex and in other parts of the brain. So one of the thoughts were that as you start to build up these short-term memories that you are then storing as long-term memories, the pattern of synapses and the molecules that are strengthened in these synapses have to be mirrored or have to be sort of redesigned in another area of the cortex or another area of the brain. And somehow this strengthening has to occur. Now we're going to talk a little bit about this particular process. It's thought that if this process actually occurs, and the reason why you remember the name of someone that you haven't seen in a number of years, like your 6th grade teacher or your grade 9 teacher, all of these will be permanently associated with neurons no longer in the hippocampus, but those synapses and the proteins that were in those synapses are now mirrored in these cortical neurons. At that point, that memory no longer requires the hippocampus. Again, we know some of that information from HM. We know that he had long-term memory, but no hippocampus. He was able to recall things that happened in his childhood without the hippocampus being present. So we know that long-term memories probably are stored within the cortex, and we know that this process of transferring strengthened synapses into memory, this consolidation, also requires a couple of different things. We know that protein synthesis is important. We need to make new proteins at the synapses, and we need to do this not just in the hippocampus, but also out in the cortex. We know that it probably requires, at the initial stage, enzymes like CAM kinase 2, but we know that other kinases, like CAM kinase 4, which we haven't talked about, are also required in this process. So one of the things that this looks like, and this is just a schematic diagram for what Paul Franklin and Morris Moscovich and others have thought of, is that initially, when you are starting to form a memory, a long-term memory that you want to keep, it starts off in the hippocampus, as is shown here. And the hippocampus has strengthened synapses, the LTP that we talked about in the last lecture, and it will start to take some of that information through the fimbria fornix, so these long lines that you see here connecting the different neuronal cell bodies up here in the cortex. We know that this begins to be mirrored. Over time, though, as we start to strengthen this synaptic pattern, we no longer have to have the hippocampus around. We need it for the short term, as we have started to store this memory here. If you were to go back a few days from today, and you were starting to try to remember what you learned in lecture today, you would still need this pathway to access those cortical modules. If you were able to store this for a year or a couple of years, the information from today's lecture would all exist outside of the hippocampus. There is a mirroring of the number of synapses out here in specific areas of the cortex. And eventually, if that memory is laid down and it becomes a permanent memory, those synapses now, the strengthened synapses, no longer exist within the hippocampus. The hippocampus has gone on and started to think about and remember other things, but all of those patterns now exist solely within the cortex. And this is why we think long-term memory begins in the hippocampus, and this is one model, and then the consolidation process that's protein synthesis dependent up here at the top, this actually requires cortical nodules or areas of the cortex, not the hippocampus. And eventually we don't need the hippocampus to be able to have those memories anymore. And again, this is primarily the work of Paul Franklin. I've already talked to you about Alcino Silva in the last lecture as well as Morris Moscovich and others who have contributed to this. So it's believed that when the hippocampus activates this group of neurons in the cortex, it's able to access and retrieve this memory. We don't understand all of that, by the way. So remember HM, for example, had his hippocampi bilaterally. Both sides were completely removed. In fact, when we go back and examine the histology of his brain, we know that the entire hippocampus was removed, and some scar tissue that had built up. It was impossible for him to use the hippocampus to retrieve memory, but yet he was still able to do so. So we don't understand all of the memory retrieval quite yet. So we don't know a large part of how this actually works, and people want to know how long-term memory, this permanent form of memory, is actually being laid down. And we also know that some individuals, including individuals here at U of T, believe that the hippocampus is only involved in consolidating memories that are about 10 years or less, and that other parts of the brain, things that involve things like long-term memories that are incremental, learning how to count, learning how to process words, etc., other areas like the entorhinal cortex that surrounds the hippocampus might be more important. And again, some anecdotal evidence that we talked about in the last lecture, as well as on your quiz, suggests that the entorhinal cortex, which is damaged in Alzheimer's disease, and as it's damaged in Alzheimer's disease, some of those long-term memories also start to become degraded, might suggest that this plays a role, whether in the retrieval of the memory or in the actual formation of the memory, or some other process. We're not, again, 100% sure, but the entorhinal cortex is known to require repetitive types of information as well as incremental learning. So counting by ones, for example, that's incremental. You've learned it, you no longer have to really think about it, etc. This doesn't really resolve how we actually form memory. And this has become the topic of a lot of debate in neuroscience. In fact, it's one of the biggest debates in neuroscience. So one of the things that I'd like you to make sure that you know, and this is a very straightforward question for question 1 on your quiz. It's actually available now. It should be available for you on your quiz page. If you were looking at what potentially might happen, this is the current model and the current theory, that somehow, out of all of those thousands of synapses, and tens of thousands of synapses, and millions of synapses that occur between different groups of neurons in the hippocampus, you have to be able to trace them back to the specific groups of synapses that are taking part in you remembering this particular slide versus what you were talking to your friend about outside of this classroom 10 minutes beforehand. So think about the number of synapses that were involved in doing that, and trying to think about what your friend was talking to you about, and a totally different group of synapses that are now trying to furiously take in this information here. This is not trivial. There are certain groups of synapses that were activated now, and those weren't activated when you were outside. How do we know which of those synapses are being activated now, are becoming stronger now, and how do we actually go back and mirror this snapshot of this memory that's taking place right now? So I told you there's a couple of pieces of information that we are pretty comfortable with. There's a lot that's unknown, but some of the information that we're comfortable with is that there are all sorts of mRNA molecules. These mRNA molecules, I started off a few lectures back talking about potential molecules that might be involved in long-term memory formation. mRNA was one of those molecules that I talked to you about initially. We kind of poo-pooed it at the time, but we're actually back to talking about mRNA. mRNA is enriched at all sorts of different synapses. In fact, all synapses that you look at have a large population of mRNAs. Why they're there, again, people have been speculating for a long period of time. We also know that if you want to form long-term memory, if you want to actually remember something for long periods of time, you need to actually synthesize new proteins. If I were to find the appropriate area in the cortex, and I inject it in either actinomycin D, so here's a little bit of a heads up. These two molecules that you see here, actinomycin D and cyclopexamide, are protein synthesis inhibitors. I'd like you to know that for the final exam as well as for the upcoming quiz. So question 1 involves these two molecules. We know that you can actually has been known for a while to actually block long-term memory. So we know that CAM kinase 2 doesn't exist as an mRNA. It exists as a protein. It's a functional protein. This cannot be blocked normally by cyclohexamide nor actinomycin D. That's one of the reasons why we don't think CAM kinase 2 is directly involved. I told you CAM kinase 2 might initiate it, CAM kinase 4 might finish it, but those are pre-existing proteins that are involved in long-term memory. So this led to this work where people wanted to know, what is this memory molecule? Is there an mRNA out there that as a synapse gets stronger, and you can see a synapse getting stronger here, and this synapse getting stronger here, whereas these other synapses that are nearby may not actually be growing in size, etc. What is the molecule that allows those two synapses, out of all of the thousands of other synapses on that one neuron, to actually get stronger and remember that specific information? What you learned in class versus what you were talking to your friend about outside. And this was sort of the big race to see who could find this molecule that allowed these synapses, but not their neighbors, to grow and to hold that information and eventually be transferred into the cortex. So these candidate molecules, there were a number of different criteria that people started to look at. So this current view, just again to reiterate, we have a number of these synapses that are going to be strengthened. We know that it is a protein synthesis dependent pathway. You need to know that cyclohexamide, actinomycin D, both of these can block this sort of strengthening of synapses. Also prevents the consolidation outside of the hippocampus and the cortex. So this group of this network of neurons and all of its different synapses that are associated with it is known as a memory trace or a memory engram. For every particular memory you have a memory trace or a memory engram and all of the molecules that are associated with it. So we know that if we disrupt this somehow, either with protein synthesis inhibitors, completely ablating neurons in very simple animals, and we take away a memory trace, the entire memory gets erased. So we know that this exists. In simple animals, it's been shown in simple networks within simple animals, eliminating one neuron and all of its surrounding synapses completely eliminate this memory trace or engram. And we know a little bit about that process. But this was what different individuals in sort of the mid-2000s were kind of dealing with. How do we actually explain why some synapses tend to get bigger and more strengthened and remember and retain that information versus others? And does this actually explain long-term memory? Not LTP, because that's a well-described phenomenon, but long-term memory and the formation of these engrams, these specific patterns of synapses within a neuron. So we know that LTP is really important in beginning this process. Learning and memory start with LTP. There is no doubt about that particular process. But when we want to look at long-term memory, beyond sort of one day into like a week, a month, a year, or even longer, your lifetime, that can't be LTP. Those synapses are turning over within your hippocampus all the time. The synapses that you have today are not going to be the same as the synapses that you have tomorrow, etc. So we know that that can't be the only way that this happens. So people started to examine candidate genes. It had to fit all the criteria that I just mentioned to you. It has to be protein synthesis dependent. Therefore, we have to look for some kind of mRNA that sits near a synapse that can get activated after that synapse starts to become activated. So one of the interesting things about this process was that if you do exactly the same types of experiments that we looked at earlier, so if I were to electrically stimulate the Schaffer collateral group of axons, which release glutamate, we already know that one of the things that will happen is that this synapse will become stronger in the short term through a process that looks like LTP. We know that glutamate will bind both NMDA and AMPA receptors, that the NMDA receptor in turn allows the entry of calcium, and this calcium will activate kinases. This kinase, when we looked at it, was thought to then cause the increased trafficking or the increased opening of AMPA receptors, leading to this synapse getting stronger. But that's not the end of the story. Other individuals started to examine what this other CAM kinase actually started to do. And one of the things that CAM kinase potentially could do, perhaps, was that it could signal back to the nucleus in a rapid way, and it potentially could actually cause the transcription of a gene. And I'm going to get to this protein kinase M in a second, this PKM zeta gene, which will then cause protein kinase M zeta, mRNA, to be translocated rapidly to the synapse. And this protein kinase M, if this synapse is being activated, will then be transcribed into a functional protein or translated into a functional protein. So without thinking too much about it, cyclohexamide, for example, or actinomycin D would block the conversion of the mRNA of protein kinase M to the full-length protein. And this is thought to actually cause the long-term maintenance of this synapse. This is the signal. This is the memory molecule that people were looking for. It was an interesting hypothesis. There were a lot of things that were going for this. One of the reasons why it was really attractive is that this mRNA, when people looked for it, was indeed enriched at all synapses. Every synapse potentially could get stronger. Great, because that fits our model that we're looking at. mRNA has to be there. Cyclohexamide can block its translation, and without this kinase present, that means that the long-term memory cascade might be affected. The other thing that was really attractive about this hypothesis was that they were able to generate a very specific PKM zeta pharmacological inhibitor. So this PKM zeta inhibitory peptide, which is known as ZIP, was found to actually block only PKM zeta, does not affect early LTP or anything else. No other kinases are affected. And when you block PKM zeta, and you block this particular protein using this PKM zeta inhibitory peptide known as ZIP, then you actually prevent or erase the memory. So if an animal had been taught something, and then you injected in ZIP, that memory, by blocking this PKM zeta formation, was completely erased. That animal forgot it had ever learned that particular task. This was really incredible when you started to think about it. You could erase any memory as long as you were able to find a specific group of synapses in a very, very specific way. And so this is what this is showing here at the bottom, that ZIP application can actually completely erase a memory when it is applied. And again, it's a long-term memory because you were doing this like a day outside of the LTP actually occurring. This is mainly the work, by the way, of Duden and Sachter. And some of you were asking a little bit about this literature review, and if your paper is only looking at one drug, and the drug is only examined by one group, how do you actually account for this, or how do you actually expand that data beyond that particular group? So I'm going to use that as a caveat for the time being. All of the work that I'm going to be showing you in the next 10 minutes actually revolve around the work that was done by Udall Duden as well as Todd Sachter, and they're at the Southern University of New York at Stony Brook, and they were particularly interested in this protein kinase M zeta. Again, one of the things that they looked at was a model involving the hippocampus. It makes sense in some ways for them to actually examine the hippocampus. The hippocampus is really important for spatial forms of learning as well as spatial forms of memory. And I'm going to go through their experiment in a few moments. And one of the things that they developed was this molecule known as beta-ZIP, or ZIP in that previous slide, which is the PKM zeta inhibitory peptide. It blocks this particular protein, in other words. Have I lost any of you so far? You guys are okay? So let me take you through their data twice. So they're using a hippocampal dependent form of memory. And one of the things that they're doing is they are using this rotating wheel. This rotating wheel has this little pi zone here that if the animal enters it under this red pi area, the animal will receive a foot shock. It will learn this association really quickly. This is also spatially dependent because the animal has cues around the outside to let it know when it is approaching this shock zone. So if it sees stars and it's moving away from those stars, that it can see on the side wall, then it knows that it is going to enter into this shock zone. So it is learning, it is remembering all of this. This involves LTP for sure. And if you waited long enough, could you actually demonstrate that it was a long-term form of memory, one day later? So again, keep in mind that the animals learn very, very quickly. So they learn that the time to enter, meaning they avoid going into the shock zone. So it takes longer and longer for them to actually enter into this shock zone. And then if they went back and tested 24 hours later, they still remembered, if they were injected with saline, they still remembered to stay out of this area that would give them a shock. These same, not the same animals, but a similar group of animals that learned in exactly the same way, when they were injected with beta-zip, after they were tested following the injection, they completely forgot everything that they had learned. They entered in and out of the shock zone almost as if they had never learned this before. So again, surprisingly, ZIP was able to disrupt this long-term form of memory because it will enter the shock zone almost like it was never trained before. So again, I'm going to show you this as a video, so you'll actually be able to see parts of their experiment. So to begin with, when these animals are just put in, this is the pre-training phase. There is no shock zone on. The animal is just getting accustomed to where these spatial cues are in the environment around it. There is no shock, so it will completely enter into this pie-shaped region. It is about 1/5 of the total area of the actual circle. So this is where we get the 20% random entry. And so you can see that it just goes in and out of that shock zone sort of at will. And again, we don't see the spatial cues around it, but you'll have to take my word for it that there are spatial cues that the animal will be looking for as it is doing this pre-training. So eventually, and I'm hoping in the next few seconds, we will start to see what happens when the shock zone is now on. So this is a late training. So it's already been shocked. It knows where the shock zone is. This would be the shock zone. The shock is actually on. And you can see that the animal has learned. This is trial 8. So this is the 8th trial that it's been in this wheel, that it knows that it would get shocked if it enters into this zone up here at the top. So you can see very, very clearly the animal is staying as far away from the shock zone as possible. And I'm hoping that it's really evident to all of you that it has learned to avoid this area. It's staying almost completely at 180 degrees away from it in this particular training session, almost the last training session that we would look at. So again, this is a form of learning as well as memory. There's no doubt that LTP has occurred, learning has occurred. Now, we need to know if there is any memory associated with it. This is the saline control injection. Okay, so this animal is now just getting 24 hours later. The training is done. You wait a full day. If it's long-term memory, it should remember this. The animal is injected with saline, and you can see that the animal, for the most part, avoids getting into that shock zone. And you can see that it does a pretty good job of remembering where that shock zone is. Even though there isn't, the shock isn't present for it, it still remembers where that area is supposed to be. You can see its head moving to try to figure out where it is in space to avoid that particular shock zone. Now, what would happen if you were to inject in this molecule that Todd Sachter and Udall Duden had created, this ZIP? So can it actually erase memory? And this becomes the really key point. So now, if you are blocking PKM zeta, what happens to this animal? Well, it doesn't care. It's forgotten its memory of the shock zone. It's also been trained for 8 trials. After injection of this PKM zeta inhibitor, all those memories stored in those synapses that remember where that shock zone is, they're completely gone. They've been reset. That memory has been forgotten, and that's why it's freely entering that shock zone. When scientists saw this paper published in Science in the mid-2000s, this was considered a classic, an instant classic. In fact, if you were to look at textbooks that were published in the year 2013, 2014, you would actually see that this was in neuroscience textbooks. This is indeed the memory molecule because it was so convincing. I mean, it's really hard to argue with the fact that if you were looking at these mice that were injected with ZIP, that all of that memory was gone. In fact, they continue to enter into those zones as if they had never learned this at all. Now, this is question 2. There are some things that you need to write down for question 2. And I also need you to know it for short answer questions, as well as for potentially multiple choice test questions on this diagram. There's a lot on here that I want to take you through, so please ask questions if anything is unclear for you as I take you through this diagram. This is an updated version of what we now think happens with protein, the PKM zeta. So let me start with this gene up here at the top. What is PKM zeta? Most of you will probably never have heard of PKM zeta. And PKM zeta is a variation of protein kinase C. All of you have heard of protein kinase C at some point in your undergrad biology careers, right? And protein kinase C is a particular variant. So this is protein kinase C Z, the gene. This will form protein kinase M zeta. And what makes this really unique, all protein kinases actually have two different domains associated with it. There's a regulatory domain that actually turns it on or turns off the kinase. And there's also a catalytic domain, which will actually be the kinase itself, where it can be an active enzyme. And we know that the PRKM zeta, which is derived from the PRKCZ gene, the PKM zeta only involves the catalytic region. There's no regulatory domain on it. So it's only the catalytic active domain. And we know that it gets transcribed as an mRNA. And this is actually put at every single synapse. Every synapse that you have within your neurons, every last one of them has PKM zeta, which kind of makes sense, or the mRNA for PKM zeta. Normally, this is not being translated. It has to be turned on by a signal, in other words. It's waiting to get the appropriate signal to become turned on by activity. And in the same way that we talked about before, glutamate will trigger this activity to begin this process. And one of the things that's really important about this is that as calcium comes in, and calcium is still an integral part of this process, coming through that NMDA receptor, that other enzymes potentially can be involved. It's no longer thought that CAM kinase 2 goes back and somehow causes the increased transcription of PKM zeta. We know that locally, either CAM kinase 2 or PI3 kinase, MAP kinase, protein kinase A, mTOR, or even actin might release the translational block that's on here. This only begins when this synapse is active though. As we're starting the process of generating LTP, this PKM zeta that's just been sitting there, now either through CAM kinase 2 or through protein PI3 kinase or MAP kinase will somehow be released, become translated, and again, it can still be blocked by cyclohexamide or actinomycin D, and this PKM zeta protein, as it gets formed, requires another kinase known as PDK1 to help it become fully activated. So we need PDK1, which normally exists, to help form the full protein, and this is that molecule which then allows for long-term memory to exist at this specific synapse if it has become stronger with this mechanism. One last thing that you'll need to know, especially for the final exam, these mRNAs that get transported from the nucleus down toward these different synaptic regions actually are all based on microtubule transport, which is what the MT stands for at the top. These require an intact microtubule network. These RNAs are transported down in cargo through anterograde transport mechanisms that involve kinesin-like family members, particularly kinesin-like family member number 5, or KIF5. And you need to know that it is anterogradely transported along microtubules using this kinesin-like family member known as KIF5. If we were to disrupt this somehow, or if we were to block KIF5, we would have exactly the same effect of not being able to have long-term memory formation because PKM zeta mRNAs can't get transported out to the appropriate synapses. So there are a number of different mechanisms. We'll see on question number 2. It's a very straightforward question as well, as long as you understood the important parts of this particular diagram. Yeah, Nancy.
The PDK1 protein, are they like coenzymes or does it just activate?
It activates and finishes the process of full translation of the PKM zeta protein itself. Okay, any other questions or things that I can answer for you? again, it's a good idea to go through the quiz. You're allowed to do it twice. If you want to come in and do it with me, by all means, please do so. If you need to come in on Friday, for example, I'll be happy to go through the quiz with you as well. I don't think it will take you very much time, and you can actually track back to this particular slide all the things that you'll need to be able to get full marks on question 2 on quiz 2. So a couple of other things that are really important. So Sachter's group actually, following their 2006 paper that was published in Science, which made great news and people were really excited by it. And I remember being in graduate school and being really excited by hearing about what they were doing in that particular process. And then when they finally published their paper in 2006, it was like, wow, they finally made it, because it was in the rumor mill for a very long period of time, about 2 years before they actually were able to publish it. But there are some criticisms of this that began early. As soon as they started publishing it, a number of people actually started to critique this. So there are a couple of major critiques that you might make on this. So one, they were using the hippocampus. I told you earlier, the hippocampus is probably important for those short-term memories, and some people might argue that you need the entorhinal cortex. So they're not looking at areas outside of the hippocampus. They're still looking within the hippocampus. This might just be another form of LTP. They're not looking in the cortex. They're not looking really long-term either. That's another major criticism. So one criticism, not outside the hippocampus, not in the cortex. And another major criticism, they're not really looking long-term, are they? It's like 24 hours after they have finished their training. Is 24 hours really long-term memory? A lot of scientists argue that's not long-term memory. Long-term memory is like a week, a month, etc. after the animal has stopped learning. There's another minor consideration that people were starting to become a little bit irritated by. So Sachter's group started to pump out huge numbers of papers related to PKM zeta, not surprisingly. So when you're invested in this molecule, you start publishing papers on it. For the very longest period of time, for about 3 or 4 years, they were the only lab that was actually publishing on PKM zeta. So when you're the only lab publishing on it, of course you're always going to get the same types of results. And so, or similar types of results that fit in with your particular model. So people were kind of asking, yeah, is anyone else able to reproduce your results? Can any other lab in the world actually find the same things that you are in this particular context? So this, again, was something that Todd Sachter's group were aware of, and it's one of those things that happens a lot in science. We talk about replication, we talk about reproducibility, and we talk about actually fitting the model or the paradigm that we already know about. So we know that long-term memory probably involves the cortex. They didn't look at the cortex in that paper that I just showed you. It was all related to the hippocampus. And so all of these things were, again, in their follow-up paper addressed in this particular figure. So let me tell you a little bit about this particular experiment, and then we'll take a break. So they wanted to get a really long-term form of memory. This is really hard in the animal world, because if you think about a month, for an animal that's living about 2 years, a month is actually a long period of time. It's the equivalent to about 5 to 10 years for you, for example. So if you're taking a look at what one of the model systems that they use something completely different. And this is a form of associative learning. It's not based on foot shocks or anything else, but it is also associated with something that is unpleasant for the animal. So there's a couple of really important things that you need to be aware of. So we do, again, ethically, and this is all done humanely, we do some water deprivation for a couple of days. They're on a really small amount of water, not to harm them in any ways, but to see how much water they'll drink when they have the opportunity to drink. So in this pre-training phase, we have water deprivation, and they are on very minimal amounts of water. And then we do a couple of different things to them. So one thing that is done is they are given water that contains saccharin. So again, saccharin is sweet tasting, and we can actually see, for example, these animals, when they've been water deprived, they'll actually increase the amount of water that they take in that has saccharin associated with it. They learn very quickly that saccharin is good. And we can also inject them intraperitoneally at the same time with sodium chloride, which doesn't do anything to them. It's just a control vehicle. So the other thing that they will do is they will also be given saccharin water. And in this particular case, they are learning that the saccharin water shouldn't be something that they drink, because now, when they're given free access after water deprivation of saccharin-flavored water, they're given an injection of lithium chloride. And they're given a high enough dose of lithium chloride that it makes them nauseous. So it actually causes them to vomit, throw up, they get very, very ill. So think back to the last time you threw up. What kind of food were you eating? Do you actually like eating that same food that you ate when you got really, really sick? My guess is, if you're like these rats and mice, that you will actually avoid that food because you make that strong association that the last time I ate this, I got really sick. I don't really like that food as much anymore. And so we can show that learning, and this is a very, very long period of learning, can actually show that this conditioned taste aversion is a form of learning that occurs out in the cortex. Another really important part to this. So these animals pick up on this association quickly. The next time that they are offered saccharin-flavored water and they were given an injection of lithium chloride, if they're given a choice of either water or saccharin-flavored water, which do you think the lithium chloride-injected animals will drink? They'll drink just the water, because they still remember the last time I drank that saccharin-flavored water, I got really sick, so I'll just stick to water. Normally they would choose, because it tastes better, they would choose the saccharin, but the lithium chloride makes that association for them. This form of memory lasts a very, very long period of time. So again, don't worry so much about all of the overall changes or the sizes of the graphs, but I wanted to indicate for you, especially in B, that if you were to take a look at what happens to these animals for this conditioned taste aversion. So if they learn all of this, and then you went back and you tested them 3 days later, or 7 days later, or 25 days later, the higher the bars are, the more they will actually go for the regular flavor, the regular water, not the saccharin flavored water. So they remember that the saccharin caused them to throw up if they were injected with lithium chloride. So if you were to inject those same trained animals with ZIP, they actually forget that association. They forget it after 3 days, or if you waited 7 days after this conditioned taste aversion and you injected them with ZIP, gave them free access to either water or saccharin-flavored water, they'll still pick saccharin-flavored water because they've forgotten that association. And then, most interestingly, 1 month later, 25 days later, most animals will remember, if they're just given an injection, not of ZIP, but just a control injection, they will still remember that saccharin caused them to throw up. They will avoid drinking saccharin-flavored water, but if you were to inject those animals before testing with ZIP, they forget that association. So this shows that this memory, which normally would last up to 25 days, which is a long-term memory, out in the cortex, actually can also be erased with ZIP. So this addressed some of the concerns that different people had, but not all of the concerns that people had. It's still Todd Sachter's group, and only Todd Sachter's group, that is showing that ZIP application can actually erase a memory that has been laid down. So this is the last slide that I'll go through, and I do need you to pick up on a couple of things on this slide. We know that one area of the brain that is particularly good and responsible for this conditioned taste aversion is this area here in the mouse and rat cortex, as well as this area here in humans. This is an area known as the insula, or the insular cortex. If you have a sense of disgust, or you dislike something really strongly, or you have that association with throwing up and eating something bad that disagrees with you, the insula becomes activated. If I were to tell you that your final exam is going to actually be fully cumulative, and it's going to involve like all the material from the first term, and it's going to be the hardest exam that you ever wrote here at U of T, your insula would be firing like crazy. You'd be disgusted, right? And so we know that this area in the cortex is really important. Some individuals have implicated disgust involves things like the basolateral amygdala, but for the most part, the insula seems to be the major dominant player in them. This is an area of the cortex that receives a lot of sensory input, so it makes sense that the insula is involved in taste sensation as well as aversively motivated tasks. Again, this is the area that's often associated with disgust. I would say that some individuals in the past have implicated the insular cortex with things like anxiety disorders as well as other emotional disorders, but that, again, isn't a really strong correlation. Some of you are doing things related to anxiety disorders, so I put that up there for some of you who might be interested in following up on the insula as a potential model for anxiety-related disorders or emotionally-related disorders, but for the most part, it's the disgust and the conditioned taste aversion that I want you to know is associated with this area of the insular cortex. So all of those check boxes that Todd Sachter's group weren't able to address before, it's in the cortex, it's very long-term forms of memory, past 25 days, and ZIP is showing exactly the same thing there. Again, another notch for Todd Sachter's group. Everything is going really well. PKM zeta is indeed the memory molecule, and he has a way to erase all of these memories. Okay, so I think now is a good time to take a break, and I think we'll take a much faster movement break today, and it'll just last a few minutes, so if you can just hold on for 1 second. Before we get started, how many of you have classes on Thursday or Friday mornings? Okay, both days? Like both Thursday and Friday? Okay, that's rough. So I'm going to have a sort of writing room that we'll set aside at U College. So if you want to drop in and have like a draft of your written review, so either myself and or a TA will be there, they'll go through a draft with you. It won't be this week, it will be sort of next Thursday and Friday. Potentially also like next week on Wednesday before class begins. I'll post the times and rooms. I'm hoping that if you want to, that you can come in and get a quick read through of your draft or get some suggestions on it. We'll be adjusting that room continuously. If you want to, if you know that you're going to drop in for sure, and you want to go over your test or anything else, you can also bring your test to the different rooms that we'll have for these sort of outside the regular office hours. So please keep checking online, so I'll post the rooms times. Hopefully you can make it to at least one of those different times, and we'll have those up and running starting next week, and we'll try our best to give you a hand on your literature review or give you suggestions on your literature review in the upcoming few days. So when Todd Sachter actually did experiments where he was introducing the gene PKM zeta, and he had labeled it with red fluorescent protein, which you see on the leftmost panel, 3 different groups of dendrites, and each of these represent, the arrowheads represent a spine head. And he was able to label the dendrite and spines using GFP. He was able to show that overall, not every synapse had the protein itself that was being expressed. Remember, you can't get a fluorescent mRNA, you can get a fluorescent protein. So this is at the protein level. Not every synapse has a protein associated with it. So some have just GFP, but no PKM zeta associated with it. Just as he would predict that after activity, some synapses get strengthened with PKM zeta, but not every synapse gets strengthened with PKM zeta. And again, it was just another sort of thing that he was able to check off. So for the longest period of time, between 2006 to about 2011, he was doing really well. Like, people were kind of looking at his research, looking at it in a really positive way. If you go back in the literature far enough, and you were to look at the Society for Neuroscience abstracts, not before they got fully published, but starting in about the year 2012, there were some signs that maybe this story wasn't going to be quite as good as people initially thought. There were rumors that Rick Hugener, the person that I showed you at the beginning, was developing a knockout mouse model. And in science, that's one of the things that is actually really important, to go in and test the validity of different types of experimental data. And this actually was devastating. This was actually a big paper that came out in January 2013 that showed, and it was a back-to-back series of papers. He wasn't the only person to show this. Another group that I'll show you, Robert Messing's group, also showed exactly the same thing. That if you were to knock out PKM zeta, this molecule that we've been talking about, that's implicated in being at the synapse, responding to activity, being that little tag that then gets mirrored up in the cortex, if you were to knock out this gene completely, and just this gene, that in fact there was no effect on synaptic plasticity, there's no effect on learning, and there's no effect on memory at all. So this is actually really troublesome. So I'm going to take you through some of the data, also just to re-emphasize things that hopefully now you will have a better appreciation of. What structure is shown to you in panel A, both in the wild type and knockout? Should all know this structure really well by now. What brain structure? So all of you are saying hippocampus, that's great. And it's just to show you that between the wild type and knocking out PKM zeta, there is no structural change within the hippocampus. The hippocampus still exists, still has the same circuitry, still has the same number of neurons. There is no deficit in the hippocampus structurally or in terms of the number of neurons after you knock out PKM zeta, which is indicated as the knockout. So if you were to take tissue from the wild-type animal, the wild-type mouse that has PKM zeta, and you were to take the knockout animal, you would actually be able to show that the knockout animal on a northern blot analysis for the mRNA actually does not have RNA for PKM zeta. It has a smaller band that is being recognized, but this is the band that we would want to compare between wild type and knockout. And definitely when we look at it, the RNA for PKM zeta is no longer present. Remember, this is the catalytic domain of PRKC zeta, and it's completely gone. So again, structurally, there is no change. The gene itself seems to have been knocked out. And when you go to the protein level, this was actually a really great series of Western blots that he showed. So we're really looking at the wild type on the left and the knockout animals here on the right. And if we look at PKM zeta, one isoform of it, we see it in the wild type. This is now the protein. And we see that in the knockout animal, there is no protein that is found here at all. The other isoform of PKM zeta is also completely knocked out. Both isoforms of PKM zeta that they were able to identify is completely gone. And I hope that you see on this Western blot that there is no PKM zeta. Importantly, when they did this experiment back in 2013, and you have to keep this in mind because I'm going to come back to this in about 10 minutes. All of the other isoforms that they were able to identify in 2013, so PKC lambda, for example, there is no increase nor decrease in any of the other protein kinase C isoforms. Remember, PKM zeta comes initially from protein kinase C zeta, but none of the other protein kinase C isoforms that exist within the brain, PKC lambda, PKC alpha, PKC beta, none of them are either increased nor decreased. There is no compensation, in other words, by knocking out PKM zeta. This is a very important thing to do in science, to show that there's no compensation, there's no other change. The only thing that you're changing is PKM zeta, and he's done a really great job of showing that at the RNA level, and more importantly, at the level of the protein itself. So that's just the biochemistry. If you were to actually take a look, is there any change, for example, in the overall excitation and the potential to produce long-term potentiation in these animals? So they're using a protocol here, which is known as TBS. Now you all know what TBS is. Can you tell me what that is an acronym for? So not transcranial. Theta. So this is that theta burst stimulation that we mentioned in passing in the last lecture. And this is short bursts of high frequency activity. And after they do theta burst activity, we see that both the knockout animal in the black circles and the wild type animals have LTP. Not surprising because Todd Sachter says it's not LTP, it's long-term memory. Okay, so again, the knockout animals have LTP. It looks identical to the wild type animals that you see here using theta burst stimulation. They also use what is known as HFS. Do you know what HFS is? Or can you give me another word for HFS or another description of HFS? So high frequency stimulation or 100 hertz action potentials. And they do another different type of stimuli to show that using this model, they also are able to induce LTP in both the knockout as well as the wild type. Is all of that clear for everyone here? Yes? You see LTP, you see that structurally the animals are the same, and they have indeed knocked out only protein kinase M zeta. I'm hoping that's all clear for you. So again, if you go through, this is where the wheels fall off the bus. Do you remember what ZIP is supposed to be specific for? ZIP is supposed to specifically block only protein kinase M zeta, only that enzyme. Remember, in the knockout animals, there is no protein kinase M zeta at all, period. So this is that figure shown right here. There is no PKM zeta. So they apply ZIP, and look at what happens. So when they apply ZIP in this black bar, so during this black bar, they continuously apply to that hippocampal slice 4 to 5 micromolar ZIP. And when they apply it, almost immediately after they apply it, this is what happens to the LTP. What are you seeing happening to the LTP? It goes down. It disappears. In fact, the synapse becomes weaker. It goes under 100%. Okay, maybe that happens. We know that ZIP can inhibit protein kinase PKM zeta. But here's the interesting thing. This is also done in the knockout. What is ZIP working on? And this, in the knockout animal, in the black bars sort of hidden behind it, ZIP is doing exactly the same thing. It is causing the rundown or the elimination of LTP. So this is when they apply it, and when they continually apply it, ZIP is causing the LTP to disappear. But there's no PKM zeta in these knockout animals. So how is this potentially working? So again, if you go through several other types of experimental data, and if you were to take a look, in the knockout animal, if you were to induce LTP, which is the top bars that you see up here, indeed, if you add ZIP, it causes the same phenomenon that we saw over here. It causes this rundown. More interestingly, if I were to look at other synapses, ones that aren't actually being strengthened, just any synapse between the hippocampus, the CA1, and the Schaffer collaterals, if I use a control synapse, this control synapse, when I apply ZIP, is also going down in this knockout animal. There's no PKM zeta. This is not LTP. This is just baseline. This synapse is getting weaker. What ZIP is doing is just generally causing synapses to become weaker. It has nothing to do with PKM zeta at all. So I'm hoping that you're starting to see some of that picture, especially when we look at the knockout. So does this occur in the wild type? We see exactly the same phenomenon in the wild type. Down here in panel B, we're seeing that after high-frequency stimulation on the top, we certainly see LTP, and the application of ZIP here at the bottom causes this rundown, this elimination of the strengthened synapse. But in a non-strengthened synapse, when we apply ZIP, we also see the weakening of that synapse too. So independently of what PKM zeta is supposed to be doing, it is doing something bad to a synapse. It is causing the weakening of a synapse. It probably has nothing to do with PKM zeta directly, at least based on this knockout data. You could also argue, which is what Todd Sackter's group did, is that you have really bad technique. Like, the reason why you're getting this is you just don't know how to do electrophysiological recording. I'm sorry. That the reason why you get sucky data is because you have sucky technique. And so one of the things that is done as a control, when you take a look at the wild-type animal and then you apply a non-specific PKA inhibitor, in the presence of this PKA inhibitor, do you see rundown of LTP in panel D? Does LTP disappear? It does not. So you don't expect it to, because PKA is not directly involved in LTP, so it shouldn't affect it, and they certainly don't see that. And in the non-tetanized, in the non-strengthened synapse, you also see rundown when you apply PKA5, which is the PKA inhibitor. It does not change. So it has nothing to do with technique. It has everything to do with the fact that ZIP, the molecule that they thought was specific for memory erasure, probably is just generally weakening all synapses, whether they're strengthened or non-strengthened. This was a devastating blow to to Todd Sachter's group. It was not the only paper that came out. In the same issue of Nature in January 2013, Robert Messing's group did behavioral studies showing exactly the same thing. Knocking out PKM zeta had no effect whatsoever on learning, memory retention, or anything else. I don't want to go through all of this data with you. I think I've made the point that knocking out PKM zeta has no effect on learning, memory, and zit seems to be the issue that is driving home this point. So this was not really good news for Todd Sachter. This meant that the molecule that was touted as the memory molecule, PKM zeta, being picked up by active synapses that were getting stronger, that were eventually going to help form memory engrams in the cortex, where they were going to be reproduced, could not possibly be that molecule that everyone thought was going to be so important back in 2016. So for a very long period of time, between 2013 to the late part of 2015, there was really no response from Todd Sachter's group, which again is very interesting in terms of the dynamics. Until a paper came out in 2016 from Todd Sachter's group, which actually this individual here actually was the first author on, and they were able to show that when PKM zeta is knocked out, so they got the knockout animals, they reproduced the result. If you knock out PKM zeta, there's a different protein kinase that comes back. So again, it's in its own way controversial. I won't go through all of this, nor do you need to know all of the details. If you're really interested in it, you can go read this 2016 paper in eLife. And Sokas and Todd Sachter's group, again, tries to reestablish that PKM zeta is indeed the molecule that they said it was. Do you think Rick Hugener was really happy to see this? Do you think Rick Hugener believed this paper? No. Yes, no, maybe? No. So actually, if you go through this, there's a lot when you get these types of open publications. If you go through the open publications, you actually get to see some of the reviewer comments, which is unusual. And so there is the whole issue of backchat. There's a lot of gossip going on behind the scenes in neuroscience, and a lot of this backchat suggests very strongly that Rick Hugener simply doesn't believe this story either. So this is an ongoing battle, because what I've heard through the grapevine is that other PKC molecule that they have discovered magically comes back up. He's knocking that one out too. So he's really going after Todd Sachter again, showing that this other molecule that they found that may take the place of PKM zeta or help PKM zeta, they're knocking that one out. And they're trying to show that none of this pathway is actually really that important. So who knew that neuroscience was so brutal? And that even though learning and memory is something that we study, that we still have a lot of questions related to different aspects of neuroscience. And so in many ways, looking for this memory molecule has been a lot like looking for unicorns. Might seem magical. They might exist, they might not exist. Or in this case, they probably don't exist. And so I want to take you through the last part of this course, which relates to higher belief systems. And do you think a mouse, for example, actually has a higher belief system? How many of you believe that, I shouldn't use the word believe, how many of you think a mouse actually has a higher belief system? Maybe? Okay, I'm going to try to convince you by the end of next lecture that mice actually believe in things. And I'm going to try to convince you of that using some of the arguments that I'm going to start with here today. Do any of you know this individual here up on screen? I see a lot of you have subscribed to his product, or formerly his product. I see many of you with these. You have a Microsoft website. But a lot of you have these Apple devices. And this is Steve Jobs. And this is one of those stories that actually really makes me angry, because Steve Jobs, at one point, was considered one of the smartest individuals alive on the planet. So he wasn't the inventor of the computing devices, but he was really good at making sure that people wanted his devices. And he was a really good innovator in that way. So you take this really smart person, and then you take a look at why this smart person actually fell into this trap of not doing the right thing because of the way the search for unicorns actually exists. I want you as a U of T student, and one of the things that we've covered throughout this course is to look at primary papers. You should always be reading papers. I tend to read about 4 or 5 papers every single day just because I like reading papers. I know you're not at the stage where you might enjoy reading papers, but it's important that you get in that practice, because what I tell you may or may not be true. I can tell you it's probably true. But it's up to you to decide whether you believe in the types of things that you're learning in your classes, and the only way that you can do that is to actually read the papers that are coming out in the literature. I want you to know that not every publication that comes out on Medline or on PubMed is actually a good paper. There are several examples where papers are not really all that great. And I'm going to use a couple of different things like multiple sclerosis to kind of demonstrate what I mean by this fact that some publications that might be cited are actually not incredibly good ones. And then I'm going to get into belief systems if I have time at the end of today's lecture, but I will definitely continue on in the next lecture, and then we'll get into meditating mice and what that means for mice when they start meditating. Okay, so I do need you to know the next few slides. I know it doesn't sound like it's neuroscience, but it is health related. I want you to think a little bit about this. So if you were to read through Walter Isaacson's biography of Steve Jobs that came out just shortly after his death, one of the things that you will know is that Steve Jobs passed away because he had pancreatic cancer. And if you read through the biography of Steve Jobs has everything in the world going for him. So one, he's incredibly rich. Two, he happens to be in Silicon Valley. And if you have the opportunity to study in Silicon Valley, please do so. It's an incredible place. Really, really smart people everywhere that you go. And one of the things that was really good about being in Silicon Valley is that he had full access as a rich individual to healthcare that wasn't available to anyone else. So he went to Stanford University Medical Center. He visited someone because he had pain in his abdominal cavity. Didn't know what was going on, but he felt it was really, really serious. So again, because he's in this particularly bright place, Stanford, where they have a lot of really well-trained physicians, the resident there decides that rather than just doing a routine screen, that because he's not quite in the right age range, so he's sort of in his late 50s, he's not quite in his 60s and 70s, he kind of suspects that maybe because of your abdominal pains, you might actually have a form of cancer. So he decides to actually do an ultrasound. The ultrasound comes back a little bit weird. Again, normally not done for different individuals. If you or I, for example, wanted to go in and get tested, we wouldn't normally be given an ultrasound, but he's Steve Jobs, he gets the full treatment. So he, on this ultrasound, they see a shadow on his pancreas. They decide to actually go in and do a small biopsy. And on this biopsy that they do, no one else would have had it done, but because he's rich, because he's in the right place, they actually do a biopsy of his pancreas and they find that he has pancreatic cancer, the beginning of pancreatic cancer. It's not a full-blown pancreatic cancer yet. But the good thing for Steve Jobs is that he actually has an operable form of pancreatic cancer. If he had it removed immediately, surgically, he would have had a complete recovery. Instead, he decides to rely on all of the advice that's given to him. He decides that he's going to drink carrot juice, and he's going to drink lots of antioxidants. And the antioxidants are going to stop the cancer from spreading, and that he'll make a full recovery just based on holistic natural properties. So even though the surgical option was available, and again, if you read through Walter Isaacson's book, the resident almost breaks down in tears because it's miraculous. They found it early, it's operable, they can do it right there on the spot, but he decides, I don't want to do it. And of course, he dies from pancreatic cancer and complications related to pancreatic cancer as it spread throughout the rest of his body. And that makes me incredibly angry. How do you get a smart person like Steve Jobs possibly not taking full advantage of all of these things that none of us in a similar position would have been able to get? So it's an operable form of pancreatic cancer. They're catching it early. He's at Stanford where they can do this type of surgery, and he's rich enough to be able to afford this, but he decides that he doesn't want to do all of this. And I want you to know a little bit about pancreatic cancer. It's one of those questions that you'll get on your quiz. So this is actually a really important killer. It's one of the fourth most common forms of cancer-related deaths. It's a lethal silent killer. You won't know until it's too late. Usually it hits individuals who are late in their lives, 60 to 70 years of age. And again, it's totally asymptomatic. It might be growing inside of you. You might not even be aware that it's inside of you. And it's completely asymptomatic until something goes wrong, like a stomach pain in the case of Steve Jobs. It might be in the form of unexpected weight loss, but lots of things cause unexpected weight loss. Staying up for a week to study and do all of your assignments, for example, might lead to unexpected weight loss. Lots of things cause this, but pancreatic cancer might be one of those causes. You might, as Steve Jobs had, pain in the upper abdominal region, which again, lots of things can cause pain in the upper abdominal region. He did not have jaundice or a yellow discoloration of the skin, nor did they find any gastric outlet obstruction. And this, again, for those of you who are in physiology, Trousseau was one of the great 19th century physiologists. He actually has two Trousseau signs associated with him. One is known as a tetanic form of Trousseau sign. This is not related to tetany or muscle contraction. This Trousseau sign is where your blood is hypercoagulating. It's more likely to be able to coagulate, and that's a sign in some forms of pancreatic cancer that this Trousseau sign of hypercoagulability is a sign or an indicator of pancreatic cancer as well. Since we talked about the gut microbiome a long time ago now, do any of you know what steatorrhea is? Steatorrhea. Kind of sounds like diarrhea, so you can make the implication, but it's not diarrhea. What's steatorrhea?
Siri, what is...
Sorry?
Siri, what is steatorrhea?
Don't ask Siri. Yes. Yes, fatty poo. So if you have poo that floats, it's not good for you, right? So steatorrhea, or fat in your poop, is actually also related to signs, potentially, of pancreatic cancer as well. So again, out of all of those different signs and symptoms, Steve Jobs only had one of those, but he lucked out and he got the proper diagnosis. Again, it's much more common as you get older. It's more likely as you get older. If you're a smoker, you're also more likely to develop pancreatic cancer, not just lung cancer, but pancreatic cancer is also really important. If you are on a high-fat diet or on a very high-protein diet for long periods of time, we're not talking like 1 or 2 days, we're not talking even a couple of years, but in your lifetime, if you've had a long, protracted high-fat, high-protein content diet, you will also have a predisposition of developing pancreatic cancer as well. Many of you will also know Helicobacter pylori. It's one of the choices that you get on question 4. You're trying to pick the wrong one, the one that's incorrect. So Helicobacter pylori has also been associated with pancreatic cancer. You guys all know what Helicobacter pylori is. So it's a bacteria that's believed to live within your gut, within your stomach in particular, and it seems to be immune to the digestive juices that normally would help to eliminate most bacteria. But Helicobacter pylori has been related to gastric cancer as well as pancreatic cancer as well. So other things that are common and associated with pancreatic cancer include mutations to the BRCA2 gene, as well as the PALB gene, which is the partner and localizer of BRCA2, and it's also been implicated in different forms of cancer because these are the different proteins that are important in repairing double-stranded DNA breaks. If you have a mutation there, it can result in pancreatic cancer. Again, if we were to take a look, 90% of malignant forms of pancreatic cancer are the adenocarcinoma type of pancreatic cancers. This means that there is a displacement or an abnormal proliferation of cells that are found within the ductal region. So this would be, for example, the duct that empties out the exocrine parts of the pancreas into the small intestine, or the cystic regions of the pancreas as well. So it's not the endocrine part. And the rest, which actually was what Steve Jobs had, he had a neuroendocrine form of an islet cell tumor. These normally, if treated early, can be benign, so they grow abnormally, but they normally don't cause aggressive forms of cancer that lead to death. Again, these neuroendocrine islet cells are the minority of the type. The vast majority involve the ductal cystic type of adenocarcinomas. In either case, if you are looking at the malignant forms of pancreatic cancer, this is one of those forms of cancer that, unfortunately, we haven't made a lot of good progress on. Many of you will know, for example, that many types of cancer actually have fairly good survival rates now if you're treated or if you undergo different forms of therapy. but pancreatic cancer is actually not so good. So, for example, if you are looking at a 1 year or 5 year survival rate, so if you're diagnosed today, you get treated 1 year later, how many of those patients that were operated on, what percentage of them would be surviving after 1 year and after 5 years? It's less than 50% after 1 year, and it's less than 20% after 5 years. It's not a very good prognosis. Even if you get all of the different things that are on here, even if you get the complete removal of all of the malignant cells that are found in the pancreas, complete removal of the pancreas, go through chemotherapy, go through all of the resulting therapy, afterwards, you still are not likely to survive pancreatic cancer. It is a devastating disorder. And so again, I don't need you to know the details on this. If you're in physiology, you might be interested in this modified Whipple procedure, where different parts of the digestive tract are actually removed, and then put back together again. And again, it's the only way that people can survive for a few years, but in the long run, pancreatic cancer doesn't have a good winning streak associated with it. One other thing, pancreatic cancer is actually very painful. For those of you who know individuals that have had pancreatic cancer, you'll know that it is incredibly painful. And as a result of this type of pain, usually some sort of ganglion block where portions of the ganglia are either permanently frozen or are removed. But again, this is a last-ditch resort. Because they're in so much pain, usually it's not a very good prognosis for these individuals. And again, this slide, you don't have to know all the details of what structures are removed and what structures are actually put back together in this Whipple procedure. But if you're interested, please do refer back to this slide. You don't need to know it at all for your final exam. So for me, I'm always interested in why really smart people actually take and do some really bad decisions. For Steve Jobs, that was part of his lifestyle. He had grown up in the 60s. It was something that he was used to. He was used to a naturopathic lifestyle. And for him, having surgery was sort of an option that he didn't want to discuss until he had tried everything else. And it's one of those things that even if you're getting really good advice, and one of the things that I want you to do is to come up with your own way of rationalizing what you think is good versus not so good science. You're going to be asked this all the time. If people know that you're studying here at U of T, it may not seem like a big deal to you now, but you probably have had family members who have asked, like, oh, should I take this vitamin or should I not take this vitamin? Or what do you think about this thing that was in the news? They'll ask you because they respect your knowledge and your ability to make these types of judgment calls. So I want you to start thinking a little bit about why people make these bad decisions, why we have these different types of belief systems. So there's nothing wrong with a belief system. I have my own belief systems, for example. But when an option that is evidence-based is presented to you, I think it's always in our best interest to make sure that we are taking those types of evidence-based practices that are beneficial to us. And so I want us to start looking at how we look at papers and why we look at papers the way that we do. Some of you are looking at clinical trial papers or papers that are more clinically based. Many of you will have heard, for example, of this randomized, double-blinded, and most importantly, placebo-controlled trials, these RCTs, taken as the gold standard. And I want us to understand the biology of placebos, because that's really important for us as neurobiologists, and how that impacts us now in 2017. And again, I'm not going to ask you to know any of the details here, but I am going to ask you to keep in mind this idea that nowadays, in 2017, in evidence-based practices, we always have to have some kind of control that involves a placebo. If you're given a drug, you're usually given some kind of non-efficacious inert substance as a placebo or as a control. And we want to keep this in mind, that this placebo actually has many caveats associated with it. The double-blinded stuff and everything else I'll come back to as we need to. None of these slides, by the way, are ones that you need to know. The next few slides are ones that I don't want you to focus on at all. So we know that there are a lot of different articles. Some of you are covering some of these articles related to different compounds that might be really good for Alzheimer's disease or treating Parkinson's or really good at potentially curing things like multiple sclerosis. how do you decide if that's a good paper or a not so good paper? And how do you know that some of these test compounds that are in PubMed, and they're published in PubMed, actually are worthwhile pursuing? Even if it's in PubMed, does it actually give it the legitimacy to make it a really good paper? So again, there's a lot of things that we need to start looking at. We need to look at the accuracy of some of the different reports and some of the different literature that's cited in these papers. When you read through those introductions that many of you are writing now, sometimes those references are taken completely out of context. The accuracy of the reports are actually completely wrong. Sometimes the authorities in a given field are actually not real researchers, or they're not real clinicians in the traditional sense. There's always bias. I'm biased, for example, towards evidence-based medicine. I'm aware of that. I want that to be an open discussion, that I like having proof of what I'm talking about in terms of what I share with you as students. But we all have biases, and we need to keep that in mind when we're covering and going through the literature as well. And a lot of these articles that we're talking about, so if you knew nothing else, and you went in and covered a paper on PKM zeta today, in 2013, you would think, for example, or prior to 2013, you would think that PKM zeta is the memory molecule. There's no controversy associated with it. One of the reasons why we ask you to look at current articles, science is changing all the time. What we think is right today could turn out to be completely wrong tomorrow. So we need to look at how current some of these different models, some of these different molecules, etc. are. And just like when we looked at Todd Sachter, if it's only one person publishing the same paper about the same molecule all the time, how accurate do you think that's going to be? That person also has a bias associated with that as well. And again, if you're looking at clinical trials, always look up these clinical trials on different websites to see where they're actually going, or if they've actually failed in these different clinical trials. And do they meet all of these different criteria that we'll come back and talk about a little bit more next week. So I want to just cover one particular aspect, because it is related to neuroscience. And this gets into, do we really believe all the different things about multiple sclerosis? In Canada, this is incredibly relevant for us. Canada has the highest rates of multiple sclerosis in the world. We're not talking North America. In the world, we have the highest rates. Saskatchewan, within Canada, has the highest rates of multiple sclerosis. We don't really know all of the causes that are associated with multiple sclerosis, but we're pretty good at actually picking up multiple sclerosis. And normally I don't ask you to know all of these different details. I'm going to highlight the ones that you need to know, and you're going to have to circle these. I want you to use this because you'll see this in question 5. For example, one of the signs and symptoms of multiple sclerosis are changes in vision, and that's one of the signs that often comes up. So we know, for example, that nystagmus, or the abnormal movement toward the side of your eyes, or being able to track things in space, is often associated with multiple sclerosis. If you've never had that before and you have nystagmus, a visual problem, that could be a sign of multiple sclerosis. Diplopia, another potential sign of multiple sclerosis. You might also have fatigue that you've never experienced before, or cognitive impairment for unknown reasons that you've never had before as well. You may have problems with swallowing, especially dysphagia. That's often associated with more severe forms of multiple sclerosis, or you might have problems with your speech that you've never had before, dysarthria. Again, in terms of sensation, you might also have the paresthesia, or abnormal pain. Does anyone have a sort of anecdotal or a more common term for what paresthesia is? All of you have had paresthesia at one point or another. Paresthesia. Any ideas? It's a type of pain that you have in your extremities, and people with multiple sclerosis have a lot of it. It's actually really painful for them. Have you ever had your leg fall asleep and you have these little pins and needles when the blood circulation has been restored? That's paresthesia, but they have it a lot, or they have it continually, and it's really bothersome for them, especially when it becomes a big distraction. So paresthesia is something that you will see on your quiz, and I want you to be able to know where that's coming from on this diagram. We know that multiple sclerosis is a disease of the CNS, it involves probably an autoimmune response that causes the gradual demyelination of neurons. And if it happens to be demyelination of neurons in your visual cortex, it might lead to visual problems. If it's a demyelination of neurons that are involved in the somatosensory cortex, that involves processing of pain, that might lead to paresthesia. If it's demyelination in other areas, it might account for cognitive impairment. If it's in the frontal cortex, etc. But we know that it can actually be detected on things like MRI if it's caught at certain stages. Again, we don't know a lot about the genetics. In fact, the only paper, by the way, that came out on the genetics of multiple sclerosis has actually been questioned. We don't really know if there is a genetic model for multiple sclerosis, and we don't know about the genetic susceptibility. If this is so common, and it is, then we should be able to pick up the genetics, but we have a really hard time right now of linking one gene or multiple genes to multiple sclerosis. So it might be genes and having some environmental susceptibility, but we're not 100% sure of all of this right now. We know that you can do MRIs at different times to be able to pick up certain forms of multiple sclerosis. You can also do evoked potential, so you can actually put in electrodes and see what the overall responses are in the muscle. So you can see if it's a problem with the nerves or a problem with the muscle to give you that differential diagnosis of why you might actually have weakness in your muscle. And these evoked potentials help. You can also do a lumbar puncture to look for the presence of different antibodies that are often associated with increased autoimmunity. But all of these are just guesses in the dark. We don't really know until multiple sclerosis begins to progress. So again, it's much like what we talked about with depression. So again, this is in the age range of 15 to 40. This is when it strikes the most often. It also strikes women more often than it does men. So it's about 3 times higher. We are actually one of the highest rates in Canada. We don't know if it's only due to a lack of sunlight. We don't know if it's a lack of vitamin D. These are all great suppositions. But when we go back and test out some of those in an evidence-based way, the data seems to be all over the place. Maybe it is, maybe it's not vitamin D. Maybe it's related to sunlight, maybe it's not related to sunlight. There is a clear correlation, though, between being in the northern hemisphere or farther away from direct sunlight and having an increased risk of developing multiple sclerosis. But that's all that it is right now, just a correlation. Again, 3 Canadians, which is a very high rate, 3 Canadians are diagnosed newly with multiple sclerosis every single day, and it's fairly high. It's about 1 in 500 individuals. So this isn't like one of the really big disorders of the nervous system. So if you take, for example, schizophrenia, affects 1 in 100 individuals. Multiple sclerosis affects 1 in 500. So it's not an uncommon disorder. In other words, many people that you know will have multiple sclerosis. 80% or greater actually have one particular form of multiple sclerosis, which is known as the relapsing remitting. So they'll get better for a little while, and then they'll have a bout of multiple sclerosis. They may not be able to see very well, or they may have problems with their motor coordination. It doesn't have to affect them the same way every single time. It may change the way that it presents itself depending on where the demyelination is occurring. Out of that 80% plus, more troubling is that you will go on to develop the progressive form of multiple sclerosis. This is a problem because you never get better. The relapsing and remitting, you'll have good days and bad days, but once you convert to the progressive form, it's all bad days from that point forward. You will never have a recovery day afterwards. And this becomes very, very debilitating. It definitely impacts their lifestyle. And again, as part of question 5, if you were to take a look, there is a suggestion that immune cells are involved in this process. I think I was going to ask Cortana something. I'm not sure. So there's a suggestion that it is an autoimmune disorder that might lead to a disruption of the myelin sheath around central neurons, and so it disrupts the signals that travel down these axons. And over time, as more and more of these myelin areas start to degenerate and start to become lost, these will actually result in these plaques that you see being built up that are detectable on an MRI. These can actually, it looks really bad to have these types of relapsing, remitting forms of MS plaques, but these can actually resolve, they can disappear. These can come back. The myelin, over time, can actually go back to being fairly normal, so these will disappear with time. But for some cases, this will not get better, it will become irreversible, and they won't actually be able to recover full function. There is no cure right now for MS. People are taking a variety of different immunomodulatory drugs that can help with the inflammation. Again, you don't have to memorize this list. I'd like you to make sure that you know it's potentially autoimmune. We don't have a really good cure for it. There are some treatments to help with the process. Some people believe it shortens the bout of MS when they have it, or that the symptoms aren't quite as bad if you're on some sort of interferon beta when you have that attack of multiple sclerosis. But for the most part, we don't have a really good way to prevent multiple sclerosis or to completely treat it. And again, steroids have their own side effects associated with them. Maybe they do help to reduce the time that you actually have this relapsing form. And these drugs are really there to help treat the symptoms. There's no cure available for multiple sclerosis. So I happened to meet a few individuals with multiple sclerosis who were living with it. This, again, was sort of an issue a few years back related to the bioethics of, what do we do for these individuals that really don't feel comfortable with their lifestyle the way that it is? And how bothersome is it for these individuals when they have no potential for getting better? So when you look at these types of individuals who know, because they're doing the same types of literature searches that all of you are, that there's no cure for multiple sclerosis, nor do we have a good gene or target or therapy for multiple sclerosis. Their lifestyle is really bad. They may have problems eating. They may have problems living a normal life. They may not be able to walk anymore. They may not be able to talk anymore. Their cognitive functions may be declining. Then you might almost be able to understand why people might be really interested in other forms of therapy that might work or might not work. And this actually becomes one of those gray zones. So this individual here, Paolo Zamboni, actually was a surgeon and professor at the University of Ferrara in Italy. He had a wife, Alana Ribali, who was 37 years of age. So right in that zone or that age, 15 to 40, where individuals might start to develop multiple sclerosis. She had been very healthy before that. She was married to her. And she started to develop all the classic signs and symptoms: vision loss, a lack of coordination, falling over, muscle weakness, dysarthria, inability to swallow. It got worse. And of course, as a surgeon, surgeons have a mindset of wanting to go in and be the heroes, to rescue, do things, cut things out, make changes. And of course, he wanted to do exactly the same thing for his wife. Who wouldn't want to when their wife is sick? So as a surgeon, he starts to go into the classic literature, everything he could read at the time on multiple sclerosis. And one of the things that he read, which was really interesting, suggested that there was a link between the amount of iron that was in the brains of individuals with multiple sclerosis and the severity of multiple sclerosis. So he began to think, why would there be more iron deposition that might potentially lead to oxidative, radical damage, etc. So you have iron ions like Fe2+, Fe3+, that might lead to neuronal degeneration through oxidative radical formation. So one of the things that he started to think about was maybe there's a problem with how much blood is actually draining from the brains of individuals with multiple sclerosis. That might mean that the blood is staying up there and not circulating appropriately. It might lead to more iron deposition because it's carried in the blood. And as a result, maybe this is what causes the cell loss that might be associated with multiple sclerosis. So as a surgeon, he started to use ultrasound to look at multiple sclerosis patients, and he found that in 90% of these multiple sclerosis patients, that they had abnormal blood vessels, particularly in the veins that would normally drain the brain. And so he called this finding that he was able to identify in his Italian patients chronic cerebrospinal venous insufficiency, which is often abbreviated as CCVSI or SVI. And so he tried to do a new technique surgically as a surgeon. There's no help for these individuals. There's nothing else. A lot of them want to just try anything because they want to feel better. So he decided, as a surgeon, I'm going to go in and I'm going to actually inflate the veins that drain the brain, because if I can restore circulation, maybe there'll be less iron deposition, less cell damage. And he called this the liberation therapy. How many of you have heard of the liberation technique or liberation therapy? Because it was big news a few years back. And in 73% of his patients that underwent this particular procedure, and by the way, it was not double-blinded, nor was it properly controlled at the time, he just wanted to help people. Most of them actually said, I feel better. I actually don't feel the symptoms quite as badly as I did before this procedure had taken place. So this was deemed to be a success. But of course this became really controversial because there was no cure, and particularly in Canada, where we have such a high rate of multiple sclerosis, Canadians wanted in on this. And this became a problem because Health Canada didn't want to support this therapy without more evidence to actually back up whether or not this was helpful. And so this became a problem where individuals found places outside of Canada to go and get this surgery done. And people were going down, for example, to the Caribbean to get this type of surgery done. And Mahir Mostich was one individual who actually died because the surgeons that were responsible for doing this, he had multiple sclerosis, actually weren't able to properly help him after post-surgical complications. So this became, again, a really hotbed of controversy until a University of Buffalo study, a really large study that was done with double-blinded, randomized, and a proper control where patients had the procedure done but didn't have their veins inflated or opened, showed that there was actually no benefit from having this procedure done. So we're really right back to where we began, although now we do have some more evidence to suggest that maybe this is not the technique that is all that useful. So I think I'm going to stop here. I know I have a few slides left. I know it's been a long day, so next week we'll start to wrap things up a little bit in this course. I am going to recommend, if you can, to come in at least to one of the different days, either the Wednesday, Thursday, or Friday of next week. We'll have a writing room open for you. Please bring in a draft or bring in specific questions that we can address. If you want us to bring your term test to the room that will indicate online, please let us know. I'll be happy to let you actually take your exam away with you after you've viewed it and you see that the marking is to your satisfaction. Have a great weekend. Please keep working on your literature reviews, and I'll see you all next Wednesday.